This particular subject, I had the opportunity to attend a conference at Harvard University about a year and a half ago. And one of the speakers, which I'm sure a lot of you heard of, David Sinclair, because he's very big or popular in the anti-aging world. There was 33 scientists speaking on their particular targeted subject that they've been doing research on. And David Sinclair did his subject matter on his research related to these transcription factors. There's three of them. And it was quite, for me anyway, maybe I should preface it that way, profound in terms of his research, at not only the fact that his research proves some dramatic changes in results in his laboratory studies, but also the certainty that he had, being that Harvard funds his life for, his laboratories there, his money comes from Harvard. So for him to say something that would embarrass Harvard, I don't think he would be that, I was going to use the word stupid to do, but I already did. So bottom line is, it was phenomenally fascinating for me, to the point I was excited. After I got home from that, I started doing research to find out a lot more about the subject matter, which we're going to have some discussion on tonight. And I hope you see the potential there, because unequivocally, Sinclair says, I don't think that there's a possibility that we can turn back aging factors. He says, I think it's certain that we can now. And the reason he did it is because of several studies that he presented on various types of laboratory animals and their conditions. So in one instance, there was lab animals that were blind. And by doing these three transcription factors, they restored all their sight. No stem cells used, only epigenetic information turning on these three transcription factors. Completely reversed Alzheimer's in mice with these three transcription factors. So, and there's more. So in the UK, the interesting thing is 6 months later, here comes another research study where in the UK, they use these same three transcription factors to reverse skin aging by 30 years in humans, meaning 53-year-old reverse skin aging down to a 23-year-old. And they did that, make sure you're sitting down, in 14 days. So obviously, will we be able to do it in 14 days with nutraceuticals or other frequency type devices? Maybe, maybe not. But can we do it in 6 weeks? I think we would be very happy with that, or 8 weeks, or even 90 days or whatever. But I just fascinated with the potential to know sincerely what unquestionably they are reversing. And they right now have just done a study that they're doing in humans with the eyes, which I didn't think they were going to go that fast, because I figured, okay, this really isn't going to be available to the public for another 10 years because of all the steps you have to jump through in research. But they're moving it, usually they go to monkeys or apes monkeys next. But they actually have a human study, particularly on vision, that they're recently initiated and was approved. So I think it's very exciting, and I hope that by getting the information tonight, you'll see the potential and the possibilities that all of us have or can do with some of the information and the products that we can access through Inno-Vita as well. So with that, let's get started. This is a new breakthrough in science of cellular health and longevity, which it definitely is. Like I said, Sinclair was actually making a statement. He didn't think that there was actually any true way to reverse the whole cycle that was going on with aging, because in cellular health, because of there's so many factors involved, from, as we've discussed, many of them, and Dr. Kim has created products for many of them. whether we're talking about autophagy or some of the others as well, until he came across this epigenetic transcription factors. Research discovered partial activation of these three transcription factors. Now they're called OCT4, SOX2, and KLF4. From here on out, I'm going to refer to them as SOX, meaning SOK, so I don't have to keep saying OCT4, SOX2, KLF4 every time it comes up. Could reverse aging markers without erasing cell identity, and this was a huge indicator for the scientific community. And doing some research, they didn't put the fourth transcription factor, which is associated with the Yamanowski, who discovered this in the first place. There were four of them, and they left out C-Mike, which is the one associated with activating onconogenes, which we really don't want to be doing. The foundation question: cells may not lose function permanently, but they may lose access to useful gene instruction. This represents a fundamental shift in the aging science, a concept that calls epigenetic aging. Well, we're all familiar with epigenetic aging. This kind of goes into more depth, particularly related to these three transcription factors, and it shows you how it plays a key role regarding the pathways of epigenetics in cellular health, not just aging. So this is the research. It's about one of the things that I think Sinclair was so affected emotionally in terms of profound, like he always thought everything was about chemistry, but here it was about, he learned it's about information. So research from Harvard, and again at Sinclair, DNA remains largely intact with age. What changes is gene expression. The process is known as epigenetic drift. So when the epigenetics go awry due to various things we all are aware of, from chemicals, toxins, inflammation, etc., repair genes become silenced, inflammatory genes increase, and cellular function declines, because it's all about healthy cells. What are transcription factors? Transcription factors are master control proteins. They do not build cells. They control gene expression. And this is important to differentiate what they do. They control gene expression. They don't build cells. They determine what genes turn on, what genes turn off. So this is a big deal. And most of the time they will go dormant. And when they go dormant, then the body starts losing the epigenetic factors that it needs to make the cell become a youthful cell. So the best way I could think of to put it is DNA. Think of DNA as a library. Genes are the books. Transcription factors are the librarians. So they decide which books to open, which books to remain. They determine cellular behavior. So they are the librarians is okay to go with, but they're the software, not the hardware, is the best way to think of transcription factors, whereas the DNA is more the hardware. Okay? I hope this helps differentiate what they are and what they're not. Why these three transcription factors? Because they're responsible for cell identity, repair pathways, regeneration programs, and developmental genes. So they operate at the highest level of cellular health in terms of cellular function. So that's why they're key. So they even are higher up in the epigenetic profile. I think that would be the best terms I could think of at the moment. So they're part of reprogramming factors. And again, when people like Sinclair realized that they could rewind cellular aging markers while keeping the cell function as is, that changed everything. So this was huge for the scientific community. So they're referred to as a cellular reset switch. They may help reverse. Okay, here's where, and this is on me, they may help reverse cellular aging. No, they will help reverse cellular aging and health span. So again, a key component here that you have to understand where the shift is being made, particularly among the scientific community, is this is not about wear and tear on DNA structure, but loss of information. So it's about information. So what if cells don't forget how they should be when they're young and what has to be happening? And it's based on information, which is where these transcription factors come in and the role they play with epigenetics. These cells actually became younger. I think this is what blew Sinclair away. He says the cells, when we did these studies on these laboratory animals, when set blind mice vision returns and they looked at the cells under the microscope, they were like young, healthy cells again, all through activate these transcription factors, activating proper genetic transcript or genes. And so the cells remain what they are. They don't change, where in some cases stem cells could change cells. And they function like younger versions. They identify, their identity is preserved and their functions restored. So their mechanism is resetting the epigenetic noise. And they refer to the epigenetic noise as being not a clear signal that's occurring information-wise on the cellular level that's supposed to happen in order for things to work properly. So the transcription factors help restore chromatin organization, improve gene accuracy, and reduce transcription errors. So you get cleaner cellular function. So it's a big deal when you get into epigenetics about the genetic noise or epigenetic shift. And again, because it plays a big role in the effect it has on information being transferred. They reactivate repair systems. So this is a biggie here. They help restore DNA repair genes. They help restore cellular stress response. They help restore damage recovery systems, and cells regain their maintenance ability. So, and of a lot of you that are on this and have heard the talk on sirtuins, there is a correlation between sirtuins, which I will just mention a few things, and the role they play with transcription factors as well. So they are key factors in all these things, which are what make cells be healthy and run better. So again, aging disrupts cellular coordination. So we all know, because we've heard me talk about it as well as other people, how much cellular health is the key, and what damages cellular health eventually damages health span, causing chronic health issues. And this happens because it affects signaling pathways, tissue coordination, integration, and organ health, essentially. So partial reprogramming, meaning not overstimulating, but gently stimulating the upregulation of these three transcription factors, makes it more clinical beneficial. So again, they've done several now animal studies where there's restored youthful gene expression. So that's huge. I mean, that really means, and again, I'm going on 76, guys. So obviously I'm very excited about this stuff. And the sooner that I can get my hands on well, I guess I'll let the cat out of the bag, potential product Dr. Kim's been working on to help support transcription, these transcriptions expression, that will be phenomenal. So it will truly be supporting age reversal on a cellular level. The old aging may be reversal at a cellular level without altering DNA sequence, only gene expression. So again, our traditional approach has always been not us, I would say, medically speaking, treat the symptoms and try to slow the damage. This is all about another way that we have to look at how we as clinicians can restore cellular instructions and at the same time fix root cause, and fix root cause we'll cover next. So potential applications of this is neurodegeneration, muscle loss, tissue repair, injury recovery, healthy aging support. Here's where the nutraceuticals come in. So nutraceuticals, based on the medical research, and not all of it, because now there's medical research saying that there are specific nutraceuticals that do increase the expression of these three particular transcription factors. So that's the road Dr. Kim's going down. So that's what we're interested in, besides the fact that nutraceutical helps support DNA integrity, cellular energy, epigenetic stability, repair capacity. So in other words, remember what we mentioned earlier. When we're talking about these transcription factors, we're talking about information being lost or information not getting to where it needs to go to do what it needs to do. Whereas the rest of the things we've been doing a lot with nutraceuticals is more working on the infrastructure rather than the software, if that makes sense. So these factors require proper cellular. So this is where it's saying, hey, we got to have adequate energy, DNA stability, low oxidative stress, proper NAD levels, and efficient repair systems. Well, we've already got a ton of products in terms of, from an Inno-Vita standpoint, that support all these different factors that could be suboptimal, or meaning they're actually, we want them operating at healthy levels. So by using that support creates a much better probability of us having an impact on truly increasing and or expressing those transcription factors, which we'll get into some of those products soon, which most of you will probably be familiar with. Transcription factors are the software, as I mentioned. Nutraceuticals support the hardware, the power supply, the wiring, stability, the infrastructure. So we're talking about mitochondria, we're talking about methylation, that type of thing. So the idea is at the heart of one of the most exciting discoveries, and this is, for medicine, this is the most exciting discovery in a long time. The use of these three powerful transcription factors to restore youthful cellular function is without erasing cellular identity is like considered home run, amazing. And I already mentioned about David Sinclair. And again, he is really a driving force behind this research. The research that came out in the UK on human skin, he didn't do it, but again, everybody's connecting with him since he's established himself pretty much as the expertise in these three transcription factors. So epigenetics is more associated with cellular health, is what they're saying, than genetics. Now, this is probably stepping on some people's toes. Your DNA sequence doesn't change as much as you age. What changes is how your cells read it. Over time, important genes get silenced, repair genes get turned down, stress and inflammatory genes get turned up, cellular communication degrades, and this whole process is called epigenetic drift. So obviously, epigenetic drift is a part of many factors, which we'll discuss shortly. So these three transcription factors help restore the youthful epigenetic patterns, allowing cells to behave and act like their younger version of themselves again. Like I said, on human skin, it was 30 years in 14 days that that occurred, with actually using the actual transcription factors. Okay. It improves tissue function, reverse markers of cellular aging, improves regeneration after damage, improves regeneration after damage. I mean, blind mice seeing again, that's pretty amazing. Alzheimer's mice, completely reversed, that's amazing. So, I went out and did a lot of research. I actually spent $30,000 just to create certain frequencies with an audio engineer out of Canada to build frequencies associated with those three transcription factors. And I'm thinking, I don't know what else we can do other than Dr. Kim can find nutraceuticals to express it more. So I'm going to be using the combination of the two. So this is a big deal in the scientific community, because when you're dealing on a epigenetic factor where you're doing things that help support or increase epigenetics, you want to make sure it's not turning healthy cells into or unhealthy cells even into different kinds of cells, like stem cells can be turned into other cells. So, and that's why he left out c-Myc, which is another transcription factor, but it is considered an oncogene gene, and none of us want to be turning that one on. So obviously these three transcription factors reset epigenetic noise. So they reorganize chromatin structure, which by the way, sirtuins help do that too. They restore clean gene signaling, and they reduce noise in gene expression. This allows the cells to operate with greater precision. So it's a somewhat of a new concept. As Brent mentioned earlier, it was known, the Yamanowski factors have been known for, I don't know, what was it, 10 or 15 years? Is that right, Brent? And it's been taken, and then Sinclair got a hold of it, and he ran with the ball with it, doing the research, and now it's going to be blooming everywhere in all fields of chronic diseases and health issues. And it, again, reactivates repair pathways. It reactivates repair pathways. Isn't that what we want to do after we eliminate all the various stress toxins that's causing damage? And then we want to reactivate repair pathways. And we're talking on a cellular level here. So that's how we get sick people truly healthy again. Reactivates DNA repair genes, improves cellular stress response, and restores, and I think this is a big word, resilience to damage. And of course, another biggie is they improve cellular communication. So they help cells communicate better with surrounding tissue, which I haven't found any impact that this has on the ECM extracellular matrix, particularly in fibrosis, but I'm going to see what I can find out on that. And with age, communication breaks down, as we all know. So they are referred to as the master control proteins above other transcription factors. They tell genes when to turn off, when to turn on. And so they, most aging interventions, which we've talked about, SOX works at a deeper level, much deeper level. Restoring how cells read their DNA, correcting operating system, not just symptoms. So this is why they consider the research a paradigm shift, honestly, in the scientific community. So here's the future potential benefits: restored tissue function, improved recovery, delayed onset of aging, extended health span, not just lifespan. Again, there's more recent studies coming out, and also quotes from Sinclair saying, with the research, they're saying, we can do this with liver tissue, heart tissue, kidney tissue, bone tissue, it doesn't matter. That's pretty exciting. For decades, cellular aging was considered inevitable, irreversible. The research challenges that assumption. So here it's saying, obviously, the infrastructure and is for the nutraceuticals. We're doing that with what Dr. Kim has in his toolbox, or we have in our toolbox, by addressing preserving DNA, methylation, acetylation, improving metabolic readiness, AGE, the list goes on. And we know certain plant compounds affect epigenetic markers and chromatin structure and gene expression patterns. So you have a lot already in your toolbox to support all the infrastructure around supporting these transcription factors to be their most productive. I don't know if that's the proper word to use, but so you get the most expression out of them for their benefits as to what we want, or what I want anyway. So I think that's key, and they point that out, that that's very important to do. So you have to put out anti-inflammatory, the inflammation, because it disrupts gene regulation. It disrupts epigenetic signaling, which is associated with the noise and cellular communication. And what product do you have in your toolbox from Inno-Vita is both the IntegraCell, but you also have CytoStem. So this is now changing all across the scientific community, how they're approaching cellular health, chronic health conditions, and they're now seeing that the true potential of truly restoring healthy function has a adamant possibility by activating these transcription factors. It's just we would all like to do that naturally rather than with drugs. So this is not totally a true statement in that sense. Nutraceuticals are not the switch, but they do help keep the lights on. In other words, the wiring, the whole infrastructure, as I just mentioned. But there are, and I found some studies where they know that certain, like resveratrol, vitamin C, there was a few others I found that actually increase SOX expression, which is what we want, you know. And these are the formulas that can assist in the infrastructure to help make sure we have the best transcription factor activity on a cellular level. Okay. And you go, whoa, that's a whole lot. Does that mean I have to take all of them? No, it doesn't mean you have to take all of them, but you got to have to either test or figure out which ones that individual would or should take based on their individuality. So whether we're talking about methylation, which is probably a big one, cellular sustenance, NADH, would not be surprised, to mitochond, obviously, but you test, don't guess. This all complements this whole SOX transcription factor epigenetic pathways to support it, but with the infrastructure that it has to have to function like it should in a much healthier way. Now we just need a little more support on the expression side, which the good news is, as you'll see at the end, that's something in the making. So why cellular environment matters? So you have to have adequate cellular energy. Well, we know what that is in terms of health products. Healthy epigenetic, we know what the products are. Low oxidative stress, again, we have support for that. NAD levels and DNA repair systems, we have support for that. So each one of these have specific purposes and functions on a molecular level, whether they're affecting, in this case, protein in cells. And again, remember, transcription factors are proteins. Again, you would have to test acetylation. What is acetylation? Acetylation is sort of the gas pedal, kind of the opposite of methylation. Plays a role also in gene activation and function as well. But they are the support infrastructure, power supply. We mentioned that already. Supporting NAD is critical for improved epigenetic stability, enhanced DNA repair. So out of all this, we're having this conversation about phenomenal, amazing new research that we can actually change cellular health on a reverse it in terms of turning existing cells. We're not talking about it has to make a new cell. We're talking about it can reverse existing cells back to healthy cells. So if I didn't get that point across, I need to, because this is what they're saying in the research. This is what Sinclair's saying. We're not making new cells. We're taking existing cells and reversing them by providing the necessary information that existing cells need to operate and start working again like young, healthy cells. Obviously, this is critical for infrastructure. Again, you would test, not guess. Same thing here, because these are cofactors that play roles in specific enzyme functions or specific pathways. So metabolic pathways, cellular metabolic pathways. So again, test, don't guess. So you could easily set up and that's kind of what we do, a program where you get your products and put them aside and you go, okay, we're going to test and see as far as the infrastructure supporting transcription factors. And if any of these others you need to be taking to support your activation of your transcription factors, we test these to the individual. You could, if you muscle test, you could probably test them to the specific organ. Clotho, well, you know, I'm a big advocate for Clotho because Clotho is a home run for so many things, from preventing cellular sustenance to the list goes on. It's just amazing product. Again, you can have low clotho and not have inflammation, chronic inflammation, and you can have low clotho. I mean, clotho even supports sirtuin production as well, sirtuin health as well. It's critical for the brain, heart, and kidneys. Cellular reprogramming and repair require large amounts of energy. So this is going to be mitochondria, which we all know mitochondria are associated with probably any and every chronic disease, definitely of the brain and nervous system, heart too, but probably 90% of all chronic health issues, particularly the targeted organs. So again, you would test it and see. But again, think of these as supporting the infrastructure for the expression of these particular transcription factors. And I hope that if you get anything out of this, you go, wow, this seems like a lot of detailed information about epigenetics and how's it going to benefit if you want to improve cellular health, that's how this benefits. Increasing cellular health is what? If you increase cellular health in the kidneys, you're going to have healthier kidneys. And what does that mean is less symptoms. If you increase cellular health in the eyes, you're going to have better eyes, okay? So it's critical that you look at this or at least consider it. And if you put all these supporting factors together, and then once we have his product, which I hope is going to be sooner than later, we can have another product that actually gently supports expression of these three transcription factors. Then we're in a phenomenally great position to help change our health, change our lives, and all the clientele in your life as well. Methylation, again, huge when you're talking about transcription factors and epigenetics. Autophagy, okay, if autophagy is not working, we know the cell is getting bombarded by various toxins produced from mitochondria function or other toxins that's entered the cell. So autophagy is key to support cellular health. Phosphorylation, again, another basic, fundamental affecting proteins. And again, remember, transcription factors are proteins. This is a product that I should probably be testing and using more of because of the cell membrane permeability. Both cell membrane permeability of the cell as well as cell membrane permeability of the mitochondria, because the mitochondria has cell membranes as well. This is your cellular sustenance. Now, you got to get rid of sustenance cells. We've had this conversation about cells. They do nothing but poison their environment, meaning all the other cells around them, and promote inflammation and eventually turn them into zombie cells. This is like having sustenance cells is the antichrist of health, because it will do everything to destroy everything else. So I don't know, I've not read or heard of anything where they've actually tried to inject zombie cells or sustenance cells to see if these transcription factors do anything. But I think they probably consider that as more the hopeless situation. So you want to get rid of them, period. Sirtuin, home run, got to use. I have a piece here that I didn't put in the slides because I forgot it. Sirtuins are master regulators of transcription factor behavior, acting as metabolic gatekeepers that determine whether genes are expressed in an aging or rejuvenating pattern. They do this through deacetylation, chromatin remodeling, epigenetic regulation, DNA repair coordination, and metabolic sensing, which again, if any of you say what that's about sirtuins, Dr. Kim has my sirtuin presentation, which I got a lot of very positive feedback on it. So if you need to brush up on it, sirtuins are amazing. Okay. And last statement is sirtuins, essentially a transcription fine tuner of survival pathways. So sirtuins are a biggie too. And again, you can test them, and if you do muscle testing to the specific organs or tissues, or nervous system or other pathways, and they'll soon be, we hope, a product in the near future. IntegraCell, obviously for oxidative stress, because it's listed as one of the main characters in terms of oxidative stress and damaging the infrastructure. So the transcription factors need that infrastructure in order to work properly in the epigenetic level. So key takeaway: control cellular health, restore youthful function. I mean, you literally restore youthful function. Sounds like a huge, huge promise, but I think it's really going to happen. Whether we can do the same thing with nutrition and nutraceuticals, that's the intent, and that's what I'm hoping, because we have so many things in our toolbox that are already reversing biological age. So add this to it, I can't imagine it doesn't just jump it up even a lot more. So this is a big paradigm shift, particularly in medicine, because it's taking medicine away from symptoms by looking at this. That's where the paradigm shift is. So cellular aging, cellular damage is not permanent, as they are saying now. Cells simply need to be reminded how to function young again. So it's an information issue on an epigenetic level. So here we are with the currently under development, the new product that influences gene expression to better access DNA, cellular adaptation without altering DNA. And my understanding is will support the transcription factors expression. So that's the end of my talk for tonight. I really hope I don't know if I got across the impact of these transcription factors, because it is truly a huge thing, particularly if we can increase their expression while we're supporting all those other factors. We can hit a pretty good base hit, third base hit or home run without having to go the drug route. That's my dreams and hopes and beliefs too.